So, welcome to this Meet the Expert session at this conference, the Microbe 2018 in Atlanta. This Meet the Expert session is co-organized by GARDP, the Global Antibiotic Research and Development Partnership, CARB-X, that's the Combating Antibiotic-Resistant Bacteria Initiative, and ASM. GARDP is a non-profit antibiotic drug developer, and one of their aim, in addition to these drug discovery and development activities, is to support the antibiotic R&D community with training and educational events. This Meet the Expert session is one of several events in this series. In any drug development program, safety and toxicity are main concerns and important aspects for a successful development program, but also main reasons for failure and also delays. Enable, you may have heard about Enable. It's a European public-private partnership to support early drug discovery and preclinical activities in companies and other research organizations. And they analyzed their data, and the most important reason for terminating a program was unexpected in vivo toxicology. So that shows that toxicology is really important. In the 1990s, if you remember back, PK was a main reason for a high attrition rate. Since this time, we learned to handle PK issues. We learned to use PK/PD for the best dose prediction. Since this time, toxicology has moved into the spotlight. Toxicology, we all know they are essential parts of any program with a lot of challenges. So not all animal studies are predictive for toxicology in humans. And not all toxicology problems are shown in clinical trials. And that may be an issue in the future if we have several new chemical classes coming up and entering abbreviated, shorter regulatory pathways with a smaller safety database. So we'll see what the challenges will be for toxicology. So this short introduction leads to the topic of this Meet the Expert session that has a focus on the regulatory aspects of toxicology. I would like to introduce Dr. Terry Miller. He's our expert today, and he's pharmacology toxicology supervisor at the Division of Anti-Infective Products at FDA, with long experience, more than a decade, reviewing antibiotic products at FDA. After this presentation, we will have enough time to respond to your questions regarding regulatory aspects of toxicology. So please feel free then to ask your main questions, whatever comes to your mind. With this short introduction, I would like to ask Dr. Terry Miller to come up and give us his presentation.
Thank you, Ursula. So good morning, everyone. Thank you for coming to my early morning presentation. Today I'd like to speak on the effective use of preclinical toxicology to advance antimicrobial drug development. So my name is Terry Miller, and I am the Pharmacology Toxicology Supervisor in the Division of Anti-Infective Products at CDER and FDA. Today, I would like to begin with an introduction into the non-clinical development with a toxicology perspective, describe some pharmacology toxicology requirements for a successful streamlined non-clinical program. I would like to provide some examples of issues in early and late development, and conclude with some final recommendations. As you may well know, the drug review process is a complex, multidisciplinary, stepwise process involving evaluation of both animal and human safety and efficacy data. The PharmTox reviewer in the Division of Anti-Infective Products assesses primarily the non-clinical safety data submitted by the sponsors. The efficacy data is typically reviewed by the other disciplines. So the principal focus of the toxicology review is to provide hazard identification with recommendations on clinical monitoring. So in our assessment of the non-clinical safety of a drug product, we ask ourselves the following questions. Is the non-clinical approach reasonable? Is the first-in-human starting dose safe? What are the most likely toxicities to be expected in the clinic based on the animal findings? And are these toxicity findings reversible? And what is the optimal dosing schedule and study duration in animals necessary to support clinical trials? Now, the good news is we have lots of non-clinical guidances to help guide you. The principal guidances are the ICH guidances, or the internationally negotiated guidances between the FDA, the EMA, and Japan. The guidance that we most focus on in our toxicology review is the M3R2 guidance, highlighted, bolded here. And these describe the type, duration, and timing of the non-clinical studies. And these studies, or these guidances, can be found on the FDA website at www.fda.gov. So an overall good package for a new drug application contains the following recommended non-clinical studies. They typically contain a core battery of safety pharmacology assessments, particularly studies to look at the CNS effects, respiratory, and cardiovascular systems. The package also may contain single and repeat dose toxicology studies, as well as studies to look at the genetic toxicity, reproductive toxicity, any special toxicities, such as the immunotoxicity or phototoxicity of the product, as well as carcinogenicity studies typically required for treatment durations greater than 6 months. As described in our guidance M3R2, there are certain studies required at each stage of development before the next stage of development can be initiated. For example, prior to phase 1, we typically again see a core battery safety pharmacology studies, as well as single and repeat dose toxicology studies in at least 2 species, both a rodent and a non-rodent. We typically see in vitro genotoxicity assessments, and for biologics, tissue cross-reactivity studies. Now, before initiating phase 2, we add a comprehensive gene tox core battery that includes a in vivo mammalian assessment for mutagenicity. And prior to phase 3, we continue with non-clinical evaluation of human metabolites. We add fertility and embryo-fetal development requirements, as well as studies for juvenile animal toxicity. And we also begin to address impurities and CMC issues. We do recommend that the impurities and CMC issues are addressed sooner, but we typically see them prior to start of phase 3. And at the time of the NDA submission, we then see the peri-postnatal developmental studies, which are the multi-generational reproductive toxicology studies, and again, as well as any carcinogenicity studies, if they are needed. And typically, we do receive these carcinogenicity studies as post-marketing requirements for life-threatening and serious conditions. we're typically asked the questions, what kinds of non-clinical data can sponsors rely on? We typically divide this into two separate camps. The first, for the IND, or investigational new drug application, the sponsors can typically rely on studies that they conduct, published literature, public databases, FDA drug labels. They may rely on the summary basis of approval, which are the official FDA regulatory documents for approved drugs, as well as drug monographs. Most importantly, these sources must contain sufficient detail on which to base an independent safety decision. Now, for new drug applications, it's a bit more restricted. Again, you may rely on studies that you conduct, again, published literature, and FDA drug labels. However, these labels, many of these labels are older if you rely on approved drugs, so they are reviewed for adequacy by the toxicology reviewer. The sponsors may not rely on the summary basis of approval, any FDA advisory committee materials, or non-USA regulatory assessments, such as from the EMA. For an NDA, full reports of investigation of safety are required. As I mentioned, the pivotal studies are primarily the repeat dose animal toxicology studies needed to support the early clinical trials. In our assessments, we determine whether the single dose and multiple dose studies are adequate to support the duration of the clinical trial. We look at the clinical AUC and determine whether or not the animal exposures are adequate. We ask what are the target organs that we observe in our studies, and is there a dose response for any toxic effects that are observed. We also ask if the maximal tolerated dose or the no observed adverse effect level or no observed effect level have been identified in those studies, and whether or not any of these effects are reversible. Now, there are many non-clinical challenges that are encountered in development that can delay or derail a non-clinical program. Typically, these include such things as an incorrect type and timing of the non-clinical studies to support the clinical program. The no observed adverse effect level and target organs are not identified correctly. The length of the non-clinical studies does not match the intended clinical use. This may be due to differences in half-life between species or a change in indications. Pivotal non-clinical studies may not be GLP, in compliance with GLP standards. There may be formulation issues, such as changes from the early pivotal non-clinical studies to the phase 3 clinical trials, maybe changes in the PK/ADME profile or toxicity profile, or there may be unexpected precipitation of drug in the tissue due to these formulation changes. In addition, we may see unexpected toxicities from solubilizers or other excipients. This may become important when sponsors change the formulation to add novel excipients. The overall impurities may not be qualified, such as there's a failure to identify impurity profiles in early non-clinical batches that may require additional non-clinical studies. Sponsor may have conducted insufficient reproductive toxicology battery, or we may determine at some point that carcinogenicity testing studies are needed. And again, we typically, or we can't accept these as post-marketing for serious and life-threatening disorders. To further describe some of these obstacles we've encountered in our assessments, I would like to provide some examples of some early and late developmental issues that we have seen slow a program. The first case was a new drug for a chronic indication for greater than or equal to 6 months. In this case, a 14-day dog toxicology study showed mortality which was attributed to cardiac effects and seizure. However, there was no histopathology was observed in the brain or the heart, but there were significant QTC effects. We did see tissue accumulation of the drug in the heart, the brain, and the lung at greater than 5 times plasma in this study. Before the sponsor could initiate the multiple ascending dose trial, we asked the sponsor to conduct a tissue distribution study in dogs which they showed the drug reached steady state in tissues by day 10. Because this is for a chronic indication, we also asked for a 90-day dog toxicology study at lower doses, in which a sponsor identified a NOAL or safe dose for the cardiac and neurologic toxicities. The sponsor was allowed to proceed with their trial. In this phase 1 study, subjects quickly developed CNS adverse events, and the product development was discontinued. The second case involves a novel protein that targeted a particular bacterium. 4 IV doses, once weekly to dogs, caused significant inflammation, a decrease in blood pressure, and reflex tachycardia, but only on the 3rd and the 4th dose. In this case, we suspected signs of a type 1 hypersensitivity response. In the 7-day rat and dog toxicology studies with a 3-week recovery period, we observed adventitial inflammation of the aorta, the pulmonary trunk and arteries with vascular necrosis, with very minimal reversibility. These animals showed increased heart and lung weights, damage to arterioles, especially in the lung, and again was suspected of a type 1 and type 3 hypersensitivity reaction. Before the sponsor could proceed with their clinical trial, we required an additional animal sensitization study to address our hypersensitivity concerns. The third case involved a topical gel administration using a new route and formulation of an old approved oral antibiotic for a 5-day treatment of a full thickness burn wound. Preliminary studies in a porcine animal and infected animals with full thickness burns was limited to less than 7 days, showed some evidence of efficacy, including a reduction in bacteria and reduced inflammation, and some early evidence of healing. However, these non-clinical studies used different excipients of what was proposed in the clinic. The local skin toxicity and wound healing was not adequately assessed. However, we're not concerned about the systemic toxicity of the active pharmaceutical ingredient because this product was approved for oral administration. So in this case, we required an additional GOP-compliant study in an uninfected, full-thickness burn wound model, in this case a porcine pig, with the final clinical formulation as proposed in the clinic, with administered topically to cover the full clinical duration of treatment. So in transitioning to some late development issues that we've encountered in our review of the products, the first case involved a new drug, an oral and IV antibacterial drug for an acute infection. In this case, the sponsor needed to rely on older pivotal animal studies conducted decades before with their product to qualify several product impurities late in their developmental program. However, the non-clinical lots that they needed to rely on were not adequately characterized at the time for impurities, and they caused significant emesis in the dogs, which led to uncertainties in the adequate exposure to those impurities. So in this case, before the sponsor could submit their NDA application, we required an additional 28-day rat toxicology study with enriched impurity levels to qualify the impurities at clinically relevant levels. In the second case, again, another drug, both also for oral and IV antibacterial drug for acute infection. In this case, the sponsor provided a late formulation change, which included a new excipient known to increase absorption of some drugs. They conducted a 2-week IV bridging toxicology study in rats, which showed no differences in PK or toxicology profile, which typically would be acceptable. However, it was noted that the reproductive studies were conducted with the prior formulation without the new excipient. So in this case, the sponsor for approval was required to conduct additional fetal tissue distribution study in pregnant animals, as well as an additional embryo-fetal developmental toxicology study with the new formulation. And in the third and final case, it involves an oral antimicrobial drug for a chronic parasitic infection. The sponsor conducted a standard battery of non-clinical studies submitted to support an NDA approval, which would normally be adequate. However, these studies, again, were older, and the reproductive toxicology study in question lacked an evaluation of critical developmental milestones. In this case, we required an additional reproductive toxicology study to be in alignment with current standards, and we accepted this study as a post-marketing requirement. So in conclusion, as you may know again, the non-clinical drug development process is a complex process guided by the ICH and FDA guidances and our prior experiences in the division with similar products. This process can often be streamlined with appropriate guidance from the FDA and/or toxicology consultants. It is highly recommended that you seek advice on your non-clinical development approach as early as possible. We suggest pre-IND consultation with the division, and please come ready to discuss the non-clinical approach and data that you have on hand. We also ask you consider submitting pivotal animal study protocols for review before initiating studies. We need at least approximately 6 weeks to comment on those studies at this time. Thank you. We now accept any questions you may have.
Thank you very much, Terry. So this is planned to be a very interactive session. So that's typical for a Meet the Expert session. And I see already there are urgent questions. So please, Paul, identify yourself.
Paul Perkins, Brussels, Belgium. First of all, thank you very much for your talk, very instructive and very clear. May I ask a few additional questions?
Sure.
One, what is the place that you accept for studies made on cultured cells and other cultured tissues and so non-living animal systems? What is the importance for those? Are you accepting them or are they complete just for research? What is your opinion?
So we typically, we do accept them. The official, I guess, position on the agency is that we accept them, we will review them. But at current time, they're not substitutes for the in vivo animal model. I think, my personal opinion is I think that is something that eventually will come in the future. So we do encourage sponsors to seek out and try to develop those in vitro assessments. But at this time, currently we do require the in vivo animal models, or animal studies.
Second question has to do with the GLP. Are you requiring all studies to be GLP? Because that's very difficult for universities and other non-profit organizations. We oftentimes are approached by industry to conduct some of those studies, mostly in cultured cells model, but sometimes in the animals. And of course, the universities are almost unable to do GLP studies for a number of reasons that I need not to explain, but which are fairly obvious. What is your position with those studies coming from non-GLP origin?
So according to the CFR, Title 21, Part 58, so any studies that support, pivotal studies that support the clinical trials are to be done GLP. Okay. However, we have accepted non-GLP studies in the past. What we would ask is that you describe how these studies deviate from the GLP standards and requirements. Non-GLP studies we consider supportive, and we do review those, but we have difficulty considering those pivotal.
Okay. And the last question has to do with the impurities. I really appreciate your point. I mean, impurities is an essential problem. And indeed, if we don't conduct studies and toxicological studies with impurities, we never know exactly what happens. Now, this leads me to the problem of the generics. Generics oftentimes have a certain degree of impurities, and when you analyze them in details, you find a number of things that sometimes you should not find in those ones. Are you conducting specific studies in this respect at the FDA? In other words, when an applicant comes with a generic, are you looking for impurities, including those that are not in the branded product, but are in the generic because of problems of production, problem of purifications? Sometimes you find precursors of synthesis. Sometimes you find degradation products and so forth. Just to give you an example, we looked at meropenem from different sources, and we could identify the appearance of impurities in one brand that was not present in the other brands. Are you conducting things specifically in that respect?
So we don't actually conduct any of those studies. We will assess the impurities. we have a generics group that will look at the generic products, and we consult with them closely. So they come to us if they have impurities that were detected in the generic products that are not in the innovator product. So as far as I know, those products should be identical to the branded innovator product. And if there are differences, the Center for or the Office of Generic Products will usually address that.
Yeah. The trouble is that sometimes it's difficult to find out which impurities, because if you don't know which ones to look for, you never find them. That's sometimes difficult.
True. But again, I guess the toxicologists with the generic products, they will actually obtain a profile of the generic product, and they will compare it to the branded product to see how they differ. And then the toxicologists in that group will then consult with us to see if the levels that they observe there and the novel impurities have been adequately characterized.
Okay, thank you.
Thank you.
Good morning, Barry Eisenstein. I'm CMO at Carbex, and we're pleased to be able to have you here.
Thank you.
My question is one of sensitivity, and the general question is, in your vast experience in seeing multiple products come forward, how often have you seen the animal models not predict toxicity you later saw in humans? And is there any sort of general principle or rule? Are there some species that are less good at predicting than others, for example?
That's a good question. I think in our case, what I've seen for the most part is that the animals are pretty good models for the human clinical. We don't typically ask for specific kinds of models. However, specific kind of studies, we do prefer specific models. Say for cardiovascular assessments, we prefer dogs, et cetera. So I think overall, when the toxicologist looks at those studies, a lot of times we do see a good predictability. I haven't seen too many cases where, it could have been that maybe the signal was subtle, but upon going back, we do tend to see some evidence of those findings. I know in some cases perhaps that's not the case, but I personally have not run into too many situations where the animals failed to predict at some level what the toxicity was observed in the clinic.
Hi, Jeff Ambroso, RTI International.
Mm-hmm.
A couple of questions. Regarding long half-life compounds, would you be okay with periodic dosing? So if you have a compound that accumulates and has high exposure, exposure increases over time, and it's there for quite some time, would you be okay with periodic dosing in tox studies?
Absolutely.
Okay.
We do see that somewhat often with some of our products with the long half-lives in animals. It turns out, however, that the animals tend to have shorter half-lives than people, but we do tend to see some periodic dosing in those studies.
So you're just looking to match the clinical exposure, not necessarily the dose regimen.
So in terms of the dose regimen, we would like the clinically relevant dose regimen as it's intended. However, we do know sometimes it may not be feasible to do that. So if there's an alternative dose route that provides similar bioavailability and exposure, then that's something that we have accepted.
Okay, and then another question about a non-rodent species. In some cases, I'm working with some folks who are working on compounds that are, let's say they're the fluoroquinolone class, and have a lot of cartilage effects in dogs. Is there a rationale to use monkeys versus dogs for a class like that may have a certain sensitivity?
We tend to prefer to use the most sensitive species for those, even though they are sort of these expected class effects.
Mm-hmm.
I think the preference is, in terms of our community, is to sort of use. If you can use the dogs instead of the monkeys, we generally would prefer the dogs.
Okay.
However, I think we'd feel a little bit less confident if you were to move, say, into primate species, when you know that the dog would be the sensitive species for that
Mhm.
effect.
Okay, I know in some cases fluoroquinolones, they've used dogs in early studies and then monkeys in chronic studies.
Sure.
Okay. And then maybe you could comment on Ames assays using bacteria and antibacterial agents.
Sure. Yeah, I mean, that's one of the complications we run into quite often is that we tend to have to lower the dose significantly in those Ames tests in order to see our effect. In that case, we typically would ask for an in vitro mammalian cell mutagenicity assay to sort of to go alongside with the Ames test. And then if we run into even toxicity or significant problems there, an in vivo mammalian test, like say a rodent micronucleus assay.
But the Ames is still required for antibiotics, right?
We still require the
Yeah,
Ames for antibiotics.
just lower
yes.
doses.
Except that's what it's going to be ultimately, because you're going to end up clearing the lawn. But we do still require that.
Okay, and then one final question. For compounds where you can cover the clinical AUC, but the compound is not in the systemic circulation for 24 hours, would it be sufficient to cover the AUC even though the drug is gone after, let's say, 8 hours in the animal species?
That's a good question. I think in those cases, we tend to see, again, because the animals are clearing the drug much faster, we tend to see, sometimes we ask for more than one dose, daily dose, in terms of cover not only the AUC but the Cmax. The best way we can model the clinical exposure, the better in that case.
Okay, thank you.
Thank you.
Good morning, Ursula. Terry. Terry,
Hi.
thanks. Very informative, and I think for an audience of many drug developers that is very helpful. So John Mueller, Intersys Therapeutics. I think, antibiotic developers, when it comes to reproductive tox barriers, and in the spirit of the guidance and completing that non-rodent species, I mean, rat's fundamentally primary, and then doing the non-rodent. A lot of times, as you know, that second non-rodent species is rabbit.
Yes.
And with antibiotics, we're challenged with rabbits being their sensitivity to GI toleration.
Mm-hmm.
So just wonder if you can comment for the audience, those kind of alternatives in the spirit of the guidance in meeting those parameters as, alternative species and pursuing that.
Yes. So as you mentioned, the rabbit is sometimes sensitive to the products in terms of reproductive toxicity. We still do ask that the sponsors conduct a rabbit study. A lot of times we ask because we want to see the drug taken to the maternal toxic dose, and if you can get to that maternal toxic dose, if you can get any kind of embryo fetal development effects outside of the maternal toxic dose, those may still be relevant and useful for us to review. So in terms of alternatives, we don't typically ask for any alternatives for the non-rodent, like the embryo fetal developmental study, toxicity study. We still typically see the rabbit study. But we do understand that a lot of times the rabbit, you'll hit that maternal toxic dose quite fast because of that.
Yeah, I think just specifically, when you're trying to do that preliminary non-pregnant rabbit,
Mm-hmm.
and you hit, you can't achieve a tolerated dose that is at your predicted therapeutic exposure.
Sure.
You're a little challenged on the informed nature of that study.
Sure.
So that's, I hear you, but it just presents challenges if you want to actually define margins to effect relative to your drug.
Understood. No, I absolutely agree with you. Thank you.
Here's the next question.
Good morning. Michael Atkin, Gladius Pharmaceuticals, Montreal. So I understand that formally speaking, a sponsor is only allowed to request a single pre-IND meeting with the agency. Is my understanding correct on that? And what are the rules of engagement to request informal, sort of early guidance prior to a formal pre-IND meeting?
So I'd rather not, I guess, address that myself, being a toxicologist. I have some contacts on the screen that, from our anti-infectious group. They can probably better address your question on the number of the pre-IND meetings you can have and how to request those pre-IND meetings.
Okay. Thank
Sorry,
you very much.
thank you.
May I ask also a question? I know that pediatric studies are not a main focus nowadays, but we have this big medical need for new drugs against neonatal sepsis. So what are the requirements and the juvenile toxicology studies for pediatric or neonatal programs?
So the official position is that the juvenile tox studies definitely add and contribute some valuable information. However, they are not always needed for pediatric indications. Prior to conducting juvenile tox studies, prior to determining the need for juvenile tox studies, we tend to want to look at the adult animal studies, the first-in-human or the clinical trials in patients, and then we sort of make a comprehensive decision. What do we know about the molecule? Do we expect any differences between the adult and the juvenile animals before we determine the need for a juvenile animal study? And that's kind of been the position that we've had, because there also is some limited value to a juvenile tox study, and sometimes those aren't not necessarily as informative as we would like them to be.
Thank you. Andrew Hollins in Hibrix. I was wondering if you would approach or think about potential toxicology differently if the same molecule was intended to be used in a prophylactic setting rather than a therapeutic setting.
It's a great question. Because we tend to, as I mentioned, we tend to look at as like a hazard identification. We tend to look at the overall safety. The indications, whether it be for treatment or prophylaxis, doesn't really come into play from a toxicology perspective. We want to look at, particularly say in our healthy animals, we look at what is the general safety of the product. In terms of the risk-benefit, we kind of leave that to the clinicians to determine the risk-benefit. After we inform them of all the hazards and provide our assessment of or a recommendation of clinical monitoring, they then will make that risk-benefit decision, whether it be for treatment or prophylaxis.
Thank you very much for your talk. Mark Mittenfry, Ohio State. I have a question. It's a little bit more of an in vitro focus. So thinking about QT prolongation, other cardiovascular effects, it used to be the case that we relied pretty extensively on hERG studies. Now there's a move toward more comprehensive ion channel assessment. Can you comment a little bit on the agency's position on the value of those studies?
Thank you. That's a good question. So typically, and maybe I failed to mention, typically for our first-in-human studies, we do require the hERG assay. That's still a requirement. In addition to the safety pharmacology assessment, which is usually a cardiovascular assessment conducted in dogs. Now, if we determine that there is inhibitory effects in the hERG, we sometimes do request additional ion channel studies. A lot of times the sponsor will just come with them to us anyways, because they've already begun to assess this. Because of their own concerns for cardiovascular safety. So we definitely have seen them, and on occasion we do consult with our cardiorenal divisions because they have more experience in dealing with those than we do. But we definitely do appreciate them, and we definitely do like to review them. So.
One question about the signal that should really be used to stop or just to warn people. I give you two examples. One is an antibiotic that causes phospholipidosis. You can demonstrate that in the tissue culture model, but you can also see in the animal that the compound has been brought up to the phase 3 and was stopped after that. Now, what would have been the signal to tell people you should not have gone to the phase 2, phase 3 with this compound based on the animal data? Is the lesions that you see by electron microscopy something that stops the drug? What are the requirements? What are the conditions at which you would stop the development? Take another example. Some of the compounds that I know would block the mitochondrial protein synthesis. That would be accompanied by a number of the deficiencies in the liver, but also in the bone marrow. Again, what would have been the situation that should stop the studies and should be the warning signals? Do you have anything defined, or how do you handle those things?
So for clarification, did you mention phospholipidosis? Okay, for example, phospholipidosis.
Which is sometimes said as a mild effect.
Yes.
And then the second example is mitochondrial protein synthesis, which is obviously much more important for the cell, because if you block the protein synthesis, then the mitochondria, then you lose the energy.
So the phospholipidosis is an interesting concept. From what we know about phospholipidosis is that it's a relatively benign finding in the histopathology. It's usually not associated with any kind of clinical observations. It's interesting if the study were actually stopped, or if the phase 2 trial was stopped because of phospholipidosis. It's not something typically that we would stop a study with. It may be something else associated, or it may be something that's observed concurrent or concomitant with another finding. But phospholipidosis alone is not generally something that we would be concerned about. Also because phospholipidosis is reversible. If it is truly phospholipidosis and you have determined it using electron microscopy, which is the only definitive way to see the swirls for the phospholipid vesicles, then it's likely that if with no other toxicities, I can't imagine we would stop a phase 2 trial for phospholipidosis. In terms of mitochondrial protein synthesis, that's kind of a wide-reaching it's hard to say what exact toxicities you might observe with mitochondrial protein synthesis, like inhibitors. I imagine if you're going to stop a trial for something like that, I imagine the cells that are most that require a lot of mitochondrial activity, mitochondrial protein synthesis, are usually like neurons and hepatocytes and such. So you might begin to see some neurologic findings like ataxia and whatnot. And if you see those kinds of neurological findings, those kinds of findings are typically something which we would put a hold on the trial if we begin to see those. Other findings like hepatic or kidney findings, liver kidney findings, those findings are a little bit more easily monitored. Unless it's significant, I can't imagine that you would put a trial on hold unless they were severe, because those are toxicities that are more easily monitored in the clinic. CNS effects are not easily monitored in the clinic, and for that respect, I think that would be something that would drive a full hold on a trial.
Another question. So how does the agency determine the starting dose for first-in-man studies?
Thank you. So how we typically look at the starting doses, of course the sponsor will propose their starting dose for their clinical trial. What we typically do then is we look at the animal studies, the repeat dose toxicology studies, which are the pivotal studies. And we look at the no-AL dose, or what we consider the safe dose in those studies, the studies where we don't see any adverse events. We then convert that no-AL dose to a human equivalent dose, and we have a guidance for that, on how to do that. We then compare that human dose, the human equivalent dose, or the safe dose, to what the sponsor proposed. We take that information back to our clinicians, our clinical microbiologists, and our clinical pharmacologists to see if whether or not what the sponsor proposes is adequate, or whether we want to propose a reduction in that dose, and whether or not there still be some benefit for that trial. We make a collective decision at that point on the best dose of which to recommend to the sponsor.
Are there any more questions? If not, then a last one, maybe. So if you in a program cannot determine the maximum tolerable dose, how do you determine your maximum dose?
Thank you. So in the guidance, our M3R2 guidance, it discusses limit doses. And in this case, not every study that's conducted will you need to take to a maximum tolerable dose. For studies, we typically look at a limit dose of 1,000 milligram per kilogram, which is a fairly high dose in animals. For human drugs that treat above a gram for a human drug, we typically increase that limit dose to 2,000 milligram per kilogram in animals. But alternatively, you can look at 50 times the clinical AUC is also an acceptable limit dose for a study. So again, you don't always have to take the dose to a maximum tolerable dose as long as you have those appropriate safety margins in place.
Are there more questions? So I would like to take this opportunity to tell you about the webinar series of GARDP. I've put here some flyers with a web address, and you will find this recording for this session on the website, and we will also have a new webinar where this recording will be used as basis, and you can have your online questions. So if you can think of more problems, you will have a second opportunity to ask these questions to our expert, Terry Miller from FDA. So I think that toxicology is a very important topic, and the regulatory aspects are one of them, because many programs are terminated before it comes to the agency. So we'll have also more of these webinars regarding toxicology in the future. If you're interested, please come to the drug development conference in September in Lisbon. You find also some flyers here where we will cover many aspects of antibiotic R&D. With this, I would like to thank Terry Miller for coming to this session. I would like to thank the audience for all these interesting questions, and I wish you a more successful conference the next 2 days. Thank you.